So, Pete, so why do we need reference barcodes? Why not just, what's wrong with the libraries we have?
Yeah, well, the key thing is that if we want to use the barcode data for applications, so for instance, if you have invasive species that have been detected in a port, if you have endangered species in trade, and you want to be able to enforce wildlife protection, one can't just use the data that are sitting uncurated in public systems for legislative enforcement. One needs confidence in the data. So this curation of reference libraries is critically important. High quality, tightly curated reference libraries to support legislative enforcement and trust in the data.
So you get examples of where two related species have very different ecosystem service delivery or very different impacts on an ecosystem, and distinguishing those two is really important. So you can't just use clusters. I'm just thinking about where a pollinator is endangered but its sister species isn't.
Exactly. So being absolutely confident of the identification to whether a species is common or rare, benign or harmful, and of course closely related species can have these very different attributes.
Cool.
So that's thinking about reference barcodes. What about reference genomes? Why do we need reference genomes?
Yeah, so we're in this project, we're not inventing the whole field of genomics for biodiversity, but our focus on reference genomes is that by doing a reference genome where we capture everything in the genome, everything becomes accessible for questioning. So there are lots of examples where, by understanding the diversity between species, or even within a species at the genomic level, you capture much more of the biology. I have examples from work we've done on nematodes, for example, where the diversity within the genome really tells us what's under selection in these nematodes, which are parasites in the host. So it tells us where we might focus our science in working on better vaccines or better drugs, so we can actually use the genomes for doing that. And if we do it on genome skims or just barcodes, we don't see the whole story. We're interested in speciation, so the difference between species and these two mythical pollinators, which are different. We can tell from the genomes that they're different species, but we can also think about how they've become separate species and how things are moving apart as species. And you don't get that from a non-reference genome. You have to work at the whole genome level.
Okay, so it's the completeness is critically important.
Absolutely. And you start understand the difference between what is part of the genome of the organism and what is an accessory. One of the problems with a lot of genomes that exist at the moment is they contain contamination from other sources. And by doing a reference genome where we're absolutely sure this is the beginning and the end of every chromosome, we can say this is part of the genome of the organism and not a contaminant. And so really talk about that organism and the other species that live in or on it. Its microbiome, its parasites and so on.